Are you the relative?
Yeah.
Your relative was in a major accident and he needs a new liver because it was ruptured.
We have them for rent.
That's the thing. Right now, the wait list is 3 months, and he needs one right now. But there's another option. Embryonic stem cells were first derived by 2 research groups, one headed by Gail Martin and the other one headed by Evans and Kaufman, back in 1981. The embryonic stem cells that Martin, Evans, and Kaufman derived were mouse embryonic stem cells, but we weren't able to derive the human embryonic stem cells until 1998. This was done by James Thompson at the University of Wisconsin at Madison. Now enough about the history. Let me explain to you what they actually are. Human embryonic stem cells are the building blocks of humans. They have enough information in them to build a human. They can divide and differentiate into all the systems of the human body. Embryonic stem cells, used for research or health reasons, were derived from in vitro fertilization. Embryonic stem cells are from the inner cell mass of the blastocyst. These cells have the potential to grow indefinitely without differentiating to provide millions of embryonic stem cells, which can then be directed to differentiate into any of the adult cells, including hepatocytes or liver cells. They can then grow into a functional liver for your relative. Now, this characteristic is present in embryonic stem cells because of a chemical called leukemia inhibitory factor, which allows it to self-renew without differentiating. The 2 most important properties of human embryonic stem cells is pluripotency, which means it can form any cell in the human body, and self-renewal, meaning it can continue to divide and grow without differentiating, to provide enough stem cells to make a stem cell line. To derive these cells, it's pretty tedious. You have to remove the cells from the inner cell mass and let them grow in a lab dish with a supporting medium. Once the dishes become crowded with undifferentiated stem cells, they are transferred to a fresh new dish. The process of transferring cells from a crowded dish to a new dish is called a passage. And after some time, and many passages, they could have many stem cells, even in the millions. We need to make sure that these cells that you have are still stem cells, so there are some tests that you need to perform. You first check for the self-renewal property by allowing the cells to grow in subculture for many months. If they continue to divide and grow, then you know that they still have this property. Then you check to make sure that they can still form all the cells of the human body by directed differentiation. If the stem cells can't be directed to form a particular type of cell, then you know that it isn't an embryonic stem cell anymore. Now, some other things that you check for on these supposed stem cells are surface markers. The most important one known so far is the protein OCT4 transcription factor, which is important for the self-renewal property of the undifferentiated stem cell. Another thing that needs to be done is examining the chromosomes. The number of chromosomes has to be right, and also there can be no damage to these chromosomes if you want it to differentiate properly. That way you'll be able to use them for regenerative medicine. Some other possible benefits of embryonic stem cells, besides cell-based therapies, are that we can better understand the events that occur during human development. We can also better understand the process of differentiation and how cells become specialized. Another possible benefit is human stem cells could be used to test new drugs. Testing these new drugs on differentiated cells generated from certain pluripotent cell lines means we wouldn't have to test them on humans. Back to the story. The patient ended up being alright. He had the liver transplant and everything's going fine now.